Okay, this is basically the third lecture in 231. This is the lecture on tissues. Now, the thing I always try to tell you folks is don't freak out by this chapter, because what this is an introduction to tissues. And we will look at all the different types of tissues and all the different characteristics and functions of the different tissues, but you will not have to identify tissues, really. Because as we go through the 12 organ systems or so, we'll come back. When we get to skin, we'll look at the skin under the microscope. We'll look at the sweat glands under the microscope. We'll look at everything. Then when we get to the bones, we'll look at bone under the microscope and so forth. Okay? So that doesn't mean you can just blow it off. You really do need to understand these basic concepts of tissues, but please know that if it seems confusing, it does to every student that's sitting in the seat you're sitting. Okay? Like Kyle said, look at all the slides. You got to look at them. The slides that we use on the practical exam, and we will take slides out of the slide boxes. We don't have a secret cache of slides. We'll put a slide on the microscope. We'll focus it. We'll set the light right. We'll put an arrow on something and say, what type of epithelium is this? Or what type of tissue is this? But believe me, you'll be able to answer those questions based just on these characteristics that we're going to talk about now. Okay? And I guess we're ready to go. Another one of my great pictures from my time on a ship, a Navy ship. That is a helicopter taking off of the flight deck at night. And they used to have those kind of lights like ambulances used to have that rotated. And so it rotates as the helicopter lifts off. That's a cool picture, doesn't it? Okay, how do we learn? I've talked to you a little bit about this before, and I beat this into you. If you read it in a lecture, if you hear it in a lecture, you might remember 5% of it. If you read it, maybe 10%. If you see it in an audio-visual format, which is what I like to think these PowerPoints are, you might learn 20% of it. If you see a demonstration of it, we're talking lab here, you may learn 40% of it. If you talk about it with your classmates and your lab partners, you may learn 50%. If you do a hands-on project like dissecting, you might learn 75% of it. But if you can teach it, to your lab mates, if you can teach it to your classmates, you've got a 90% retention. So interacting between yourselves and teaching each other in lab is so important. Okay? And I think you heard Kyle say that. And in fact, every time I've had a student come back and speak to another following class, there's two things they always say. They say, use the MyAMP stuff, the online stuff, and they always say, work together, work with other folks, question each other, drill each other, practice with each other, all that stuff is so important. Okay, here we go. There are 4 types of tissues. There are 4 main types of tissues. And on the exam, I can just tell you that there will be a bunch of questions, and I will say, what type of tissue is this? And what that will mean is, what is the main type of tissue this is? And there's only 4 answers, so it can't be that tough. It's like multiple choice. You know it's going to be one of 4 things, right? What are they? I'm going to actually write this on the board. Epithelial tissue. Now, what do you think that means? Skin. Well, skin is one example of epithelial tissue, but there's a lot more.
Superficial.
Yeah. Well, glands are included. But think of it as either on the outside layer of something, or the inside layer of something. So it's epithelium out here, but it's also epithelium in there. It's also epithelium lining your pharynx. It's epithelium lines your trachea and your esophagus. Epithelium lines your intestine, all the way down to your rectum. Okay? epithelium lines or covers everything. So, all your blood vessels are lined with an epithelium. Okay? Anything that's hollow, the pleura is an epithelium because it's covering. You know, the pleura is a serous membrane we talked about. It's one of these epitheliums that we'll talk about here. Okay. The other example is, I'm going to just say lines outside or inside, and then one kind of special bunch of epitheliums are your glands. All right, now how does that work? Well, think of it like this. You started off, say with your the back of your throat, and it had an epithelium on the inner lining of it here, okay? And then it goes to this, and it starts building up more layers of that epithelium. And then it goes to this, and it builds up even more and more layers like that. And then eventually it goes to this and sort of starts lining this thing in here, and it's got layers of tissues, and eventually these tissues kind of break off, and now you've got this. Okay? This is your thyroid gland, okay? Or all your glands. So, basically, all of your glands in your body form from the epithelium nearby, okay? And they get thicker and thicker, and then they break off, and then they're separate. So here's now the epithelium, and here's the thyroid gland, which isn't touching your throat anymore, okay? All right, so keep that in mind. All right, so here we go back to this. Four main types of tissues. The first one is epithelial. It covers exposed things, it lines anything internal as well, and it also forms the glands. So that's the three places you find it, which is a lot. The second type of connective tissue is called connective tissue. Connective tissue is stuff that fills internal spaces. It fills in the spaces between other organs. So it also holds us together, and it also includes things like fat. It also insulates us, stores energy. It's also things like blood. You don't really think about blood as being a tissue, but it is. Blood is a tissue, and it's a connective tissue, and it fills in the spaces between things. It also tends to transport a lot of stuff. Connective tissues also support things. So bone and cartilage are both connective tissues. Fills internal spaces. Okay, that's the second one. The third one is muscle tissue. I worked last night for about 10 minutes trying to fix this slide, trying to make this muscle tissue go over there. So it'd be 2, 3, it'd be out here, 4. I couldn't do it. Sometimes I'm a computer idiot. But anyway, so muscle tissue, muscle makes up a big part of our bodies. Obviously, muscle is what we eat when we eat a piece of meat. Meat is nothing but muscle. Okay? Sometimes there's a little cartilage in it. Sometimes there's some blood vessels and nerves running through it. You pulled those out, haven't you? Just think about what you're eating. And we're going to learn that there's 3 different types of muscle tissue. But all muscle tissue does is contract. This muscle contracts to make my arm do this. This muscle contracts to make my arm do that. Our heart, all the time, it keeps contracting in order to pump our blood around. And smooth muscle is, that's what it means here when it it says the walls of hollow organs. Think about all of you right now. You're sitting here, you're digesting that wonderful breakfast you had this morning. Right. But you know, you got muscles in the walls of your intestine that are kind of squeezing stuff on down. And they're squeezing like this just to kind of mix it all up. And that's an example of muscles in the walls of hollow organs. And there's also muscle in the wall of your breathing tubes. Anybody ever seen anybody have an asthma attack or ever have a kid that's had an asthma attack? What's it like?
They doesn't breathe very well.
They can't breathe very well. You know why? You know why they can't breathe? It's because their breathing tubes all contract. And so they won't let the air in and they won't let the air out. And it's caused by their bronchial tubes have muscle in them too, and it contracts down and they can't breathe. Okay, so it's good and bad, right? And the final one, number 4, is neural tissue. Neural tissue means the nerve cells. Most of all of that is found in your brain and in your spinal cord, but all the other nerves that carry messages out to your fingers and bring messages back from your fingers. You know, when you touch something hot, it sends a message back up to your brain that says, wow, that was hot. And then your brain says, wow, that was hot. And it sends another message back down through a muscle nerve that says, ooh, and pulls your arm back from it. But remember that neural tissue, and I'll just unload this on you right now. Your brain is nothing but a series of wires that carry electric shocks from one to the other. And it's hard to believe, because that's all it is, that we can actually think. I can look out at this room and realize that I'm looking at a bunch of faces. And the reason I recognize that as a face is because I've seen a lot of faces in my life, and all of you have too. So when you see a face, it kind of fits a pattern, and that pattern is a pattern of electric shocks in your brain that says, yeah, I've seen that before, and I recognize that as a face. Let me take it one more step back. If you're a baby, how many of you have had babies? You got babies in the family? Watching a baby, I think, is one of the most incredible things you ever get to do. Watch a little 3, 4-month-old, 5-month-old baby, and they're probably making kind of noises, and they're also touching, and they're touching their face, and they're touching their lips, and sometimes they'll touch each other, and then occasionally they'll go, because what they're doing is they're establishing those patterns. The more they touch their face, the more they recognize that something is touching their face. But they also recognize that, wait a minute, something's touching my face, and wait a minute, it's my hand. Because every time they touch their face, they're feeling it in their hand, right? So they're establishing those patterns as well. Every time they go goo-goo, they're not only saying goo-goo and establishing those patterns, but they're also hearing it. They're hearing goo-goo, and they're thinking, I'm hearing something. You know, eventually those patterns get established as well. So after a few months of doing this and touching their face about 150,000 times, they finally realize that, wait a minute, this is my face. And, oh yeah, that is my arm. And they have, fibers in their elbows and in their wrist that they finally learn that, well, wait a minute, whenever my arm is bent like that, I'm getting messages that tell me my elbow's bent. And eventually you get to where you can do this. When you get to be really old, you can close your eyes and you can do this. How did I do that? Well, it's because I have all these proprioceptive fibers in my arms and joints and everything that tell me exactly where my hand is. It's also how I can do like this and not even look at it and know that I have 2 fingers up or 3 fingers up. Did I get it right? Okay. That's one of the most amazing things about brains and neural systems and life that I just find fascinating. And how many repetitions it takes until we can do anything. You know, we have to look at a lot of faces before we can recognize faces. The number one face we look at is Mama's. You know, all of us probably looked at Mama's face 10,000 times before we could recognize it as a face. But clearly it's the one face we recognize the most. All right, epithelial. We're going to go back to number 1. We're going to spend the rest of the day and most of tomorrow on just epithelial. Epithelial are layers of cells that cover the internal or cover the external or internal surfaces of everything. The glands are things that are made from epithelial tissues that produce secretions. Glands are every one of them in the body is derived from epithelia, usually by this process of invagination, which I tried to illustrate for you poorly over here. Okay, the epithelium gets thick, it sort of invaginates, then it pinches off, and that becomes the glands. What are the characteristics of epithelia? This is important, and I'll just tell you that there's 5 of them basically. And basically they are epithelial. If we're going to create something that's going to cover this table, for example, or cover our skin, for example, it's going to take a bunch of skin cells, and those skin cells are going to have to hold together. Each one of them is going to have to connect tightly to the one next to it. And so we have these things called cell junctions, and indeed with skin and most of these epitheliums, it's a pretty tight junction that happens between cells. Another characteristic of epithelia is it always has polarity. Now that doesn't mean like it's positive or negative charged. It just means that it has a top and it has a bottom. Okay? So the top on your skin is the apical surface or the superficial surface. The bottom layer of those skin cells is called the basilar surface. Okay, the basal surface. All epitheliums are attached to whatever's below them. And that attachment is called the basal lamina. In other words, where the basal side of the cell attaches to something underneath it. And usually that thing that's underneath it is called the basal lamina. I know these are just words to you right now, but once we start looking at a few, they'll make a lot of sense. Epithelium is always avascular. So even in our skin, which our skin can be pretty thick, the epithelium, there are no blood vessels in our skin. But wait a minute, Dr. Sessions, I cut myself once and it bled. Well, that's true, because that's what you did was not just cut the epithelium, you cut all the way down through the layers below that. You cut down into the fat or the subcutaneous tissues, and there is blood vessels down there. Okay, but I'll bet every one of you at some time has had a paper cut. Oh, it hurts like crazy, and it doesn't bleed a drop. Why? Because you didn't cut all the way down through the epithelia, and the epithelia always is avascular. In every place that epithelium is found in the body, it is always avascular. So in the epithelial layers, you will never see little blood vessels wandering through there. And lastly, epithelia can regenerate. Thank goodness. Otherwise, every time we lost a little piece of it, it would never heal, right? So skin and all epithelia can regenerate. It's another characteristic. This is kind of a fake epithelial cell. It's kind of a cell that represents a lot of different types in order for us to learn some of these basic concepts. So here's the 2 surfaces. This surface down here is called the basal surface. They say basolateral, but think of this out here as the basal surface, and this up here as the apical surface. Okay, so this is exposed to whatever it's lining. And in between there's a bunch of organelles that you learned about, like mitochondria, like a nucleus, like a Golgi apparatus. And then up on the surface, up on the apical surface, this top surface, we have two variations that occur. They don't usually occur in the same cells, they occur in different cells. But some cells have microvilli. Microvilli are little bulges, and they're designed to increase the surface area so that there's more room to absorb stuff. So like if this piece of epithelium was on the inside of your stomach, or on let's say the inside of your small intestine, which is where we absorb most of our food, there would be tons of these microvilli up here that would allow for us to absorb more food, more sugar, more protein, more amino acids, all that stuff. Because that's the only way we get it, is it has to be absorbed right through this epithelium to get into the body, right? And unfortunately, our body is very efficient. If we put it in here, it's going to go on here. We absorb everything that we eat. The only thing we don't absorb that we eat is, anybody? Cellulose, that's right. Cellulose is fiber. It's the stuff that makes trees and makes leaves stiff. We cannot absorb it. We can't break it down enough to absorb it. Now, if we were cows and we had 3 stomachs or 5 stomachs or whatever they got, they can break it down. The really reason they break it down is because they have a whole bunch of bacteria in their stomach, and the bacteria break the cellulose down, and then they can absorb it. But we humans can't. And cellulose fiber is very good for us. I can promise you as a surgeon that it seemed like a third of the people that came into my office some weeks had butt problems because they were constipated all the time, and they had to bear down all the time, and they got hemorrhoids, and they got rectal tears, and they got all kinds of problems down there. And every time I would try like hell not to tell them they didn't need an operation. All they needed to do was take their Metamucil every day. If you take enough fiber, it keeps your bowels moving, keeps your stools big, which is what you want, and it keeps them soft, and it doesn't cause all kinds of problems like that. So for those of you that are in the crowd that are constipated, take your Metamucil every day. Start with a big heaping teaspoon a day, mix it in a bunch of water, drink it right down, and that's the end of it. If after a week or so you're not having nice regular bowel movements, take 2, or 1 in the morning, 1 in the evening. But if you put enough fiber into your system, remember that fiber is going to go in at the top and it's going to come out at the bottom. It actually never becomes a part of you, but it keeps everything working. My dad spent a quarter of his 92 years sitting on the toilet because he was told as a kid that you have to have a bowel movement every day. Well, you don't have to have a bowel movement every day. You just have to have a bowel movement every time you need one. Make sense? And so don't worry about it. But if they are hard and they're causing you pain, then take more fiber. You're going to get all kinds of little hints like that through the journal. Some of them will drive you crazy. Okay, so what is the function of these epithelial tissues? Well, like on the skin, they provide protection for us. They control the permeability. What does that mean, permeability? Well, it includes what things get in and what things get out. And if you don't think skin is important, go to a burn unit sometime and see a patient that has a burn over, say, even 3% of their body. If you don't have skin on a piece of skin, say this big, that piece of missing skin is continually weeping fluid. By the quart, it just leaks out. And when it comes out, it has all this good protein and sugar and all this stuff in there that then you have to remanufacture. And that's why burns are such a terrible injury, because everything has to be replaced, and they can't control what goes out because they have no epithelium. Yeah?
Also heat management.
Absolutely. That's right. Because the water is coming out, it's evaporating, it's cooling. In the burn unit, this is a lot of years ago, we used to keep the temperature at about 90. All the patients were loving it. All the staff hated it. But that's what they had to, we had to keep the rooms that warm, or else patients would get hypothermic and actually get cold.
Immunology too, get sick easily because they've got no barrier.
Right. So the skin is an important barrier. Now, probably the most important thing for these two reasons. Also, if you want to go feeling anybody, you got to have skin in order to have sensation. Right? Touchy-feely. And also, our skin makes specialized secretions. We have glandular epithelium. We sweat. We get rid of sodium. Sweat is salty, right? And that's why runners need to drink not only water, but they also, if they really sweat in a tremendous amount, probably need some salt along with it. I think I'll stop right there because it's time. All right, so we will start up there next time, and see you in lab at noon, and we'll do our little rat deal. I think as long as you have one lab book per group, you'll be just fine. And I think, like Kyle suggested, they had one person that would read the book, one person that would go get all the stuff, one person that would do the dissection, whatever, and then all work together, exchange the information. What? I don't require any homework in this class at all. I don't have you turn in anything. What? Nope. This is college. This is not junior high school. I tell you what you need to learn. You got to learn it. If it makes you feel better to do all those questions in the lab book, do them. It's a good way to learn this stuff. But I'm not going to. I don't want to have to grade it. That's the bottom line. You did? Refresh my memory. Okay, your name is? Okay. Yeah. What do you mean you can't get it till 20? You mean the bookstore won't have one till 20? Okay. You can probably go down to Palace Book and pick one up right now. I don't know what to tell you, but I know that you can get them in a bunch of places. The student association does a book trade thing. I don't know if that's happened yet, or if they still have books over there.
They don't have that book out there.
They don't have it.
I was gonna tell you if you need it really fast. I did find it like on the Kindle Fire app, like the app.
Okay.
It's cheaper, but you can rent it, and not rent it, but you can actually buy it. But it won't be a physical book. I don't know how you feel about that.
Well, what I did was I got it off of Amazon, but it's like I even expedited shipping, and the earliest I'm gonna get it is the 25th because I didn't buy my book early because I was having a hard time getting into this class and I didn't know what book to get. So basically, I guess I'm just gonna.
Well, another thing you could do is,
Yeah.
If anybody has
Just
their
give it
book,
back to me at the
ask
end
them
of
if
the
you could
term.
scan it.
Yeah.
Yeah.
Yeah, just go, because I've gotten books off for serve in the library, and then I just go scan it and stick it on a flash drive, and you could do that until your book gets here. I just don't have my book on me. I would have been like, here.
Yeah,
I
don't
would.
want to touch it right now.
Oh, no. Yeah, you don't want to.
Did you get is it a brand new one?
Alright.
Yeah, I'd give it Okay, I think we got to slide number 8. We didn't get very far into this chapter the other day, and we'll kick it from here and see. And maybe I'm wrong. Maybe we did get a little farther than this, but I think this is about right. So epithelial tissues. Remember, there's 4 kinds of tissues, and they are epithelial, connective, muscular, and nervous. Okay? So we're going to spend, I bet we spend more time on epithelial than the other 4 combined, just so you know. It seems like this is going to go on forever. It really doesn't. It gets quicker. So what are the functions of epithelial tissue? Remember, they're on the outside of everything. They're on the inside of everything. And they provide us protection. They control permeability. They allow what goes out and what comes in. They provide sensation. That's how we feel through our skin. And they provide specialized secretions. There's lots of glandular epithelium. Now remember, glands are also epithelial tissue. That's where they came from. They arose from epithelial tissue. So, but within epithelium, there's even some glands right there in the epithelium. For example, sweat glands. And in your trachea, there's mucus glands that make mucus for you to snort up. Many of the different epithelial tissues that we'll look at are specialized, and that's because they have specialized functions. So, example would be the ones like protection. Not letting fluids come in, letting them slide over and not go in would be an example. Obviously, skin is an epithelial tissue. At least the outer layer of skin is epithelial tissue. Epithelium is in lots of places that probably right now you wouldn't think about it being. For example, it's in the kidney. There's these little tubules that run through the kidney, back and forth, back and forth, back and forth. And those are long tubes. But the inner lining of those tubes is made of epithelial tissue, and the idea of it is that it allows the urine, basically what we're making as urine, to flow right through it, and it sorts out the good stuff from the bad stuff. So it's very important in permeability. It allows some things to go through it, and allows some things not to go through it. We get rid of the bad stuff, we hang on to all the good stuff in the long run. And as I said, they also make secretions, which we talked about. The polarity thing, remember we mentioned that the other day. The apical surface will often have microvilli on it. Microvilli are just little ups and downs, and what that does is it gives you a lot more room to absorb stuff through. So like on the inner lining of your intestine, where you want all that stuff that's flowing through your intestine, you want it to all be able to get up next to the surface of the epithelial tissues so that so we can absorb the good stuff out of it. So we can absorb those sugars and carbohydrates and everything we eat. So we have microvilli on the inside. We also have these things called cilia. The cilia are different than microvilli. Cilia, they move. So there are a whole bunch of little extensions there, but their job is to move. And the best place, I think, for me to describe a place that has cilia is on the inside of your trachea, your breathing tube, your trachea, your bronchial tubes that go down into your lungs. We have a whole lot of cilia in there, and that's so if as we breathed in something, if we missed catching something either with our nose hairs or one of the other defense mechanisms that we've got, then we have the cilia to catch it and push it back out. So the inner lining of our trachea has mucus cells in it, and that traps any dust that maybe made it all the way down into our lungs. But then those cilia are all working together to push stuff up the trachea. And when it gets it up the trachea, it comes into our mouth. We swallow that gross stuff, it goes down into our stomach, and it doesn't live there in our lungs. Now, why is that important? And why is that really important? I'm not going to ask you to raise hands, but if there's anybody in here that smokes, let me just tell you what cigarette smoke does to your cilia. Every time you take a puff of cigarette smoke, it paralyzes the cilia on the inside of your trachea for 12 hours. Now, I don't know too many people that smoke less than 2 cigarettes a day, because if you smoke one more often than every 12 hours, your cilia are basically paralyzed all the time. And that's why every single person that smokes, most people don't get cancer, but everybody who smokes gets chronic obstructive pulmonary disease. Because for a lifetime of not having your cilia work, a whole bunch of crap collects down in your lungs that you just have to cough up. So you'll see these old geezers sitting around at the VA hospital with a cup in their hand, and they're coughing up yellow crap out of their lungs. And I never forget interviewing these guys. I say, well, how much stuff do you cough up a day? He said, oh, about 5 cups. 5 cups. I say, and how many cigarettes do you smoke? He said, well, I guess I started when I was about 14 and smoke about 5 packs a day. And I want to ask him, have you ever taken a breath of clean air? And, it's like, well, not really. I light one right off the tip of the other one, and off I go. So, and if you're going to smoke 5 packs a day, that's pretty much what you have to do. So, these cilia are very, very important to us. And we'll show you slides of people with COPD. Chronic obstructive pulmonary disease, which is kind of a combination of all the lung diseases. Basically the emphysema and chronic bronchitis. Chronic bronchitis is all the crap that collects down in your lungs. Yeah?
So for those of like people who doesn't smoke but have a coughing problem, what is that related?
It can be lots of other things. We cough for lots of reasons.
No, my sister has been coughing for a long time. I would say 2 weeks.
Your son?
My sister,
Uh-huh.
and I don't know what.
She probably has a little upper respiratory infection, either a virus or something. I can't tell you. So anyway, one of the things that epithelial cells it's really important that they do is they maintain the integrity. In other words, there's not any big holes in them. And because of that, all of the cells, cell next to cell, they're all attached very tightly by what the book calls intercellular connections. Okay. Well, your book goes into about 3 pages, it seems to me, about these tight junctions. Good for them. I just think you just need to know that epithelial cells are attached tightly, but the great extent that the book goes into it, I think, is real overkill. So don't worry much about that. So not only are they attached together, but they have to be attached to the basal lamina, which is the basement membrane of what they attach to. And they obviously play a role in maintenance and repair. If you lose a little piece of epithelia, you got to grow it back. I've already mentioned endocrine glands, or the glands. I said glands originally originated with epithelial tissues, and they got thicker and thicker, and then they kind of pinched off and became separate as different glands. There's 2 main types of glands, and you certainly need to know these guys. One of them is called endocrine glands, and one is called exocrine glands. And the only difference between endocrine glands and exocrine glands is that endocrine glands, they're just a blob of tissue there, and they secrete their secretions. They secrete their output. They just release it out into the interstitial tissues. Interstitial tissues means tissues that are around the cells. Okay? So, like your thyroid gland, for example, it just releases thyroid hormone. It goes out into the tissues that surround all of those cells, and then it's eventually picked up by the bloodstream, and then it's taken around the body, and then those hormones act on different other organs. So, it just secretes into the interstitial fluid, this fluid around the cells. On the other hand, exocrine glands are glands that secrete into a duct. Now, a perfect example of that is the submandibular glands that live right down here below your cheek. And they have a little duct that comes up and it opens up on either side of your lingual frenulum, the little thing that holds your tongue down in the middle. There's two little holes there, and probably when you were in middle school, you got really good at gleeking, right? You can go, out would come a little squirt of spit or saliva. Well, those are exocrine glands. And lots of our glands are exocrine glands. The pancreas secretes into the pancreatic duct. The liver secretes into the common bile duct. Parotid gland empties into the mouth through a duct as well. So that's the only difference, really. Endocrine glands just secrete out into the tissues around them. Exocrine glands secrete through a duct, and that duct then empties into something. Let's talk about how do glands make secretions, okay? And I think you all understand this, although it's really pretty simple, and I don't want to make it too complicated. Basically, let's look at this cell right here, and this is an example of a merocrine gland. You need to know the difference, merocrine type A. So merocrine, the way it works is you got a nucleus down here near the basement membrane of the epithelium, and you've got Golgi apparatus. Remember, Golgi apparatus packages stuff up. It's the Federal Express of the cell, and it puts things in little packages called vesicles, and those little vesicles come up here to the surface, and the little vesicles open up and release the material that's in them. Okay? That is merocrine type of secretion. Now, it'll make more sense in a second. Example of merocrine secretion is in the salivary glands, in the spit glands. The next kind is called apocrine. Apocrine. The way apocrine glands work is they start down here at the bottom. Here's the basement membrane along the bottom. Here's the nucleus. Here's the Golgi apparatus. And the Golgi apparatus makes all these little vesicles, and all those little vesicles move up towards the apical surface of the epithelium. And then all of a sudden, it just kind of pinches off that apical part of the cell, breaks down, and this becomes all the secretion out here. So in other words, the way apocrine glands work is that they actually throw off half of the cell or so, a major part of the cell with all the secretions in it, and that's where it breaks down and goes out to do its thing. Example, mammary gland. Now, one thing I will tell you is that apocrine secretions, the actual secretions, are usually thicker. They have a lot more proteins in them, they have a lot more stuff in them than the spit glands. Remember, because spit's just like 99.5% water, okay? So merocrine is usually thinner. Apocrine glands, an example of which is the mammary gland. But notice that the cell, once it throws off that distal end, it starts regrowing and starts putting more up here, and then it throws it off again. The cell lives on. The third type of secretion is called holocrine. And the way I like to think of that is just holocrine, as in W-H-O-L-E. Holocrine, the whole cell. Okay? The way holocrine secretion works is like this. The cells fill up with all the secretions that they are manufacturing, and then the entire cell ruptures and goes out into the lumen up here. And because of that, because you're losing the cell, the whole cell is going to die as it treats itself. You've got a whole bunch of new cells continually forming down here on the basement membrane. So you notice mitosis going on here and here. We're making lots of new cells. Those cells gradually mature as they start off as really young cells here, and daughter cells, actually, 2 of them, right? And then they start picking up the secretions. They get more and more full of secretions, and eventually the cell just ruptures, and everything is lost. The best place to examine that is sebaceous glands. Sebaceous glands are basically the oil glands that you have that secrete sebum. And there's one of them attached to every single hair in your body. Okay? So we have oil glands that secrete oil here near the base of the hair shaft that keeps your hair oily. Right? And oily is good, despite the fact that the shampoo makers want you to get rid of all that oil every day. Oh, oil is terrible. Oh, but wait a minute. Let's spend another $20 and put conditioner back on it to put the oil back in, right? Just think about the logic of that. Reminds me of a story. I was sitting around the exercise club out in Dallas one time, early in the morning. I used to go out there and work out in the morning. Doesn't look like it, but I did. And we were sitting around. There was a bunch of guys. We were all drinking coffee. And there was this nurse who I'd known for 20 years. She worked on the wards, and she was a good nurse. I really liked her. And I don't know how the subject got to it. I think she brought it up, talking about shampoo. She said, oh, I just love this new kind of shampoo. And we're like, really? Is she really talking about shampoo? And we were talking about something probably important, like the football game that weekend or something. You know how it goes. Anyway, so I made the comment to her. I said, well, I have to tell you that I have never used one bottle of shampoo in my entire life. And she looked at me totally aghast, like I was from Afghanistan or something. You haven't ever used shampoo? I said, no, I have never used a bottle of shampoo in my life. She said, well, how do you clean your hair? I said, I use a bar of soap, same as I use on the rest of my body, I use it on my hair. She says, well, don't you use conditioner? I says, no, never have. And she just kept jumping on me about it. And I finally said, whoa. You use all the shampoo you want, but I don't use shampoo and never have. And I have this barber that cuts my hair about every 3 months or so. And she's a really nice lady. She's from Vietnam. And she says, oh, it's so good to see you again. You have more hair than anybody that ever comes in here to get their hair cut. You have the thickest hair. Well, there you go. If you don't use shampoo and you don't use conditioner, you'll have really healthy hair. But if you want to go out and spend all that money on shampoo and then spend more on conditioner, go right ahead. I have never used a bottle of perfume either. I do use deodorant, but not that much. Anyway. All right. What?
I just had a quick question. So on the last,
The whole print? Yeah.
It doesn't show Golgi apparatus, but is it the same?
It's still there. Yeah. Yeah. It's still manufacturing it and putting it into vesicles, and then it all goes. These little hair follicles, you probably know them best as zits, okay? Because what happens with all of our hair follicles that have a little sebaceous gland on the side of it, this little duct right here, you see this is a exocrine gland, right? Because it's secreting it into a duct that then comes out here and empties out onto the hair. This little duct gets plugged up with something, makeup, dirt, whatever, and that it doesn't have a way to tell the rest of these cells that are making this secretion to stop. So they keep on making secretion, and this thing gets clogged up and becomes a lump full of sebum, and then eventually it gets big enough that it's actually sticking out on the skin, and then sometimes it gets red and infected, and then it's red, and you gotta go in and do that business to it. I hated being a teenager, I really did. Okay. So, glandular epithelium, I've said there's basically two kinds of secretion. This is another way to split it between thin secretions, serous, think serum, think watery, and mucus, think mucus, thick, snot, all that good stuff. So these things, serous glands secrete watery secretions, mucus glands secrete mucins. Don't figure you have to put every type of gland into one of those categories and memorize it or something. Just know that thin secretions are serous, mucus secretions are thicker, and then there's some that are mixed. Well, duh. So we got some glands that secrete stuff that's kind of in between. Most cells, most glands involve a bunch of cells, like you saw in that sebaceous gland. You notice there's a bunch of cells all around here. You know, those are all individual cells, and that's pretty typical. But we do have one kind of gland that is only one cell. There's one type of unicellular gland, in other words, one-celled gland, okay? And these are the mucus cells. We also call them goblet cells, and we find them just kind of scattered out through the epithelia. So on that trachea, which we'll look at here soon, you'll see scattered little white cells that look like goblets, and those are the goblet cells. They're just one by one every so often that are making mucus. We also see a lot of them on the wall of the intestine, especially in the wall of the colon. The colon its big job is just to kind of get stuff passed on through and passed out your rectum, and it wants to be nice and slimy and slippery, so it has lots of mucus glands to keep things sliding on through you. And this is another place where I think the book goes way much into and it's crazy. Into these multicellular glands, so all the rest of the glands in our body are multicellular, except for those goblet cells that I just mentioned. And how do they organize themselves? Well, some of them are simple. In other words, it's just one long tube that has cells all along it. Some of them are divided. Let's just say, I just want to kind of familiarize yourself with these words, but please don't try to think much more of this. Tubular, obviously, is just a long, straight tube. Alveolar means it's like coming to cul-de-sacs. So there's a tube, and there's a bunch of cells around the end of it that secrete fluid, and it goes out into the street, and then into the little cul-de-sac, and then on out into the regular streets. So that's what these words mean, alveolar or acinar. We'll see a bunch of glands as we go through the body that are shaped like alveoli. The lungs, the air pockets in the lungs are alveoli for the same reason. They have a lot of little cul-de-sacs and acinar. Some of them are branched, and that's cool. But once again, don't try to memorize these or try to learn these. I just don't think it's that important. Here's a simple tubular gland. Here's a coiled gland. Here's a simple branched tubular gland. Here's a simple alveolar gland. Here's a simple branched alveolar gland. There's all kinds of them, but don't worry about any of those during memorizing the difference between them.
So the hair follicles are like
They're
coiled
more like
ones?
this.
Oh, okay.
Okay, now let's get to the naming of the epithelial tissues, because there's a whole bunch of epithelial tissues that we're going to learn here. And here's the way you name them. They all have two words. The first word is either simple, which means one cell layer, or they're stratified, which means there's a bunch of layers. Okay? So the first word of all the epithelium is either simple or it's stratified. The second word is based on the shape of the cells. They're either squamous, in which case they're all really thin. And I'll bet you looked at scrapings from the inside of your mouth under the microscope before. That's squamous cells. They're very thin. Cuboidal means just what it says. They're shaped like cubes. They're about as high as they are wide. And then there's columnar, which is usually taller, thinner, in the opposite direction as the surface. So squamous cells are thin this way, columnar cells are a little thinner this way than they are this way. There's two additional ones that you need to be aware of. You just kind of have to put those over in a different category. That's these two right here. One is called transitional epithelium. Transitional epithelium is easy because it's only found in one place, and that's in your urinary system. And the idea of transitional epithelium means that it can go from being really full, and your bladder being really distended and full, to really thin when it empties out. And so I think of transitional epithelium as sort of the retractile epithelium. It gets bigger and it gets smaller. Okay? I'll show you examples of it. The last one that's different is called pseudostratified. Now, pseudostratified, well, that means it's not really stratified, right? But it looks like it's stratified. So it's kind of pseudo. It's trying to fake you out that it's stratified, but it's not. I'll show you examples of all of these here in a minute. Okay. So here is an example of simple squamous epithelium. And by the way, and this is an example of stratified squamous epithelium. You see it's in a bunch of layers. But you may look at that epithelium and say, oh, wait a minute. These cells down here, they don't look like they're stratified. They're cuboidal, right? One more rule. You always name the epithelium by the shape of the cells that are at the top, okay? So it doesn't matter if they're cuboidal or columnar down here below. If it's stratified, in other words, there's a bunch of layers, you always name it by the layer at the top, okay? And this is pretty typical, especially like for our skin. It's real cuboidal down here at the bottom, but by the time it works its way up to the surface, stuff that flakes off, it's very thin and squamous. So squamous cells are flat and thin. Cuboidals are about cuboidal. This draws them as nice little hexagons. They're not really hexagons most of the time, but they did that just for illustration. Columnar, the big thing about columnar is they're a good bit taller, like columns. Anybody ever gone rafting out on the Deschutes River or even driven up the Columbia River and you notice the big columns of basalt, the lava, as it cooled, it cooled it into columns. I think that's always so cool to look at. All right, simple squamous epithelium. We're starting with number one. Usually it looks like this. You notice here's the basement membrane or basal lamina. Either word means the same. These are simple. There's only one cell layer thick, and they're squamous. Easy enough. Here's some examples of it under the microscope. Notice cells, single nuclei, usually out in the middle somewhere. This almost looks like it's stratified, doesn't it? Because there's one here and one there. Like there's maybe more than one layer, but don't let that fake you out too bad. When we go to lab and we start looking at these different epitheliums, you're going to find that some of the epitheliums don't seem like they belong where they are. This is an example of a slice of the kidney. Okay? This is sliced through the kidney. This is actually a glomerulus of the kidney. These are a bunch of blood vessels over here. But then there's this little sac that kind of runs around the outside of the glomerulus, and it is aligned on the outside here by a layer of simple squamous epithelium. Okay? The way we make urine is we pump blood through these capillaries here, and a lot of the fluid leaks out into this space here, and then the outside of that space, which is called the capsular space, we'll get to that later obviously, is lined by simple squamous epithelium. We call this Bowman's capsule. Be sure and look at those under the microscope. Pretty cool. Another place that simple squamous epithelium is always found is on the inside of all the blood vessels in the body. It doesn't matter whether it's the aorta, which is this big around, or whether it's a capillary, which is small enough to allow only one red cell at a time to work its way through. They're all lined by simple squamous epithelium. Now, just look at that picture for a second, and let me tell you that this is why this first unit, this chapter 4 is so confusing. Because, you know, here you are looking at a slide, but it's a tissue, remember? And tissues mean there's a whole bunch of kinds of cells in there. So these are all blood cells out here. They're in the blood vessel. This is just serum, clear fluid that's part of your blood. Here's the simple squamous epitheliums that line it. And then what's this stuff out here? This is some kind of glandular tissue that's surrounding it here. And they look like simple cuboidals to me that are out there. But the point is, when we show you these slides, you just focus on the one kind of cell we're trying to show you in there. And don't, at this point of your accumulating this knowledge, feel like you have to be able to identify every tissue that's in there. What are the functions of this simple squamous epithelium? Well, one place that it's found is lining the inside of the lung. Every alveolus, every little air sac in the lung is lined by simple squamous epithelium. Why? Well, because the cells are thin, okay? And because they're thin, they allow things to diffuse through them very quickly. So you take a deeper breath, in comes the oxygen. That oxygen immediately goes across those membranes and goes into your bloodstream. You breathe out, and that same blood which was just giving off its oxygen is now collecting CO2, and you're blowing CO2 out. So in comes the good air, the oxygen, out goes the bad air, the carbon dioxide, and round it goes. But the fact is, it happens very quickly, because you don't really take a deep breath and then hold it 10 seconds to allow that diffusion to take place, do you? It's kind of in and out, and very, very quickly. And that blood is flowing by there really quickly all at the same time, so it happens quickly. Filtration, a lot of in the kidney, those capillaries that I just showed you, that were in the glob of Bowman's capsule over there, they're very thin, they're simple squamous, and that's to allow that fluid to squeeze right out through the cells and make urine. It's not really urine at that point, but it's going to be. Another thing is that simple squamous epithelium lubricates. Remember the first day we talked about the pleural space and the peritoneal space? Well, those are simple squamous epithelium, but usually they have some mucus cells in there with them, some goblet cells, to secrete a little bit of mucus so that those layers can slide back and forth over each other, and that's its job. It's basically to lubricate. Okay. Simple cuboidal epithelium, the next one. Remember that it's a single layer, otherwise it wouldn't be called simple, right? It's cuboidal because the cells are cuboidal kind of shaped, and there they are. Every cell touches the basement membrane, and every cell touches the lumen up here at the apical surface. Okay? By definition, it's a simple type of epithelium. Here's another one. This is another area of the kidney other than the one we were looking at. But these are the convoluted tubules that occur in the kidney, and once again, we'll come back to these in great depth when we get to the chapter on kidneys. And so that filtrate that we saw created back at Bowman's capsule, it does this all through the kidneys and runs through all these convoluted tubules. They're called convoluted because that's just what they are. They're very convoluted, and there's lots and lots of tubules. So you see a slice through the kidney, and you're seeing a slice through a whole bunch of those tubes. This one may be coming this way, this one may be going that way, and so forth. Notice that all of those tubules are lined with simple cuboidal epithelium. All of these, 1, 2, 3, 4, those are all cuboidal cells. And every one of them is a simple cell because you see it go. This is the basement membrane around the outside here of these tubules, and then here's the lumen in the middle here, And so every one of these cells goes from the basement membrane out to the lumen. This is a beautiful picture. I just love this picture. I don't know why. If that's not the most beautiful, simple cuboidal epithelium you've ever seen, you'll just have to bring me a slide that looks better than that. That is so nice. This is the tubules in the kidney, an example of it. Once again, here's the basement membrane around the outside of it. These are the simple cuboidal cells in the middle. This is the lumen out here. Please notice that between the tubules, there's a bunch of other things. These are the interstitial tissues. This is connective tissue out here. And if you look carefully, I think you'll find there's some blood cells. Some of these are that's a little capillary. It has a blood cell in it. So that's pretty common. You know, you're going to see lots of other tissues around every time you're looking at any tissue. So what's the function of these simple cuboidal epitheliums? Well, a lot of glands are simple cuboidal. For example, the thyroid gland. I'll show you some pictures of thyroid gland in a minute. It's a beautiful example of simple cuboidal epithelium. They secrete hormones. They in the kidney, in those tubules that we were just looking at, they absorb simple things and they excrete simple things. I like to think of them as the real thin cells, like the squamous cells. I like to think of them as just acting as filters, almost like a screen. They let all the little stuff through and they keep the big stuff back. But the cuboidal cells are the ones that have all kinds of chemical mechanisms of pumps and so forth that pump the sodium in, pump the sodium out, pump the glucose in, pump the glucose out, and so forth. And in order to have all those pumps and stuff, they need to be thicker. So they're cuboidal. Make sense? It does in my twisted mind, at least. So anyway, that's the absorption part down there. Next one, third one, simple columnar epithelium. There are several places we'll talk about where you find simple columnar epithelium. And I would tell you, you need to know some examples of each one of these. You know, you need to know that, simple squamous is found on the inside of your mouth, usually, and simple cuboidal is found in the tubules of the kidney, things like that. But I'll just tell you right up front that simple columnar epithelium is found all the way through our gastrointestinal tract, from our stomach to our rectum. Okay? Everything between our stomach, our duodenum, small intestine, large intestine, the whole length is simple columnar epithelium. Goblet cells are often found in this layer. I'm going to show you some examples in a minute. And remember, we already said that the apical surface oftentimes will have microvilli. And in the respiratory system, they will have cilia, those things that are washing all the junk up out of your breathing tubes. So here's an example of a simple columnar. This is an example in the parotid gland. The parotid gland is one of your spit glands right here. It's the big one. John McCain, guy ran for president last time, has had cancer of the parotid gland. And so if you look at him, he's kind of got a dent in his face over here. Anyway, that's what the cells look like. These are the ducts that run through there. I'll just leave it at that. These are single cells going from the basement membrane to the lumen out here. Here's an example of I believe this is the stomach. The stomach is really thrown up into big villi, which are big folds of mucosa. And so there's all these little holes kind of down through. The food can get stuck down in here, but it makes for lots and lots of surface area. And just remember that these cells, each one of these cells then has a bunch of microvilli on it too, to make even more surface area. So this is simple columnar. So here's the basement membrane running right down here for this surface right here. Every one of these is a simple columnar cell.
Can you do that on this side? Because I couldn't see.
You can't see through me?
No. I I wish I had X-ray vision.
Yeah. All right. Well, here's the basement membrane right here. Here's the lumen out here, and it's one cell in between there. Those are all simple columnar. You see, they're kind of tall.
Yeah, like a rectangle.
Yeah. Now this picture also is nice because it illustrates the only kind of unicellular glands that we have in the body. Remember, unicellular, one-celled glands. See these kind of round whitish cells every so often? Those are goblet cells. They really look very characteristic, and as you start looking at slides in lab, they'll just jump out at you usually, even better than they show in this picture maybe. But they just secrete mucus. Inner lining of your stomach is completely covered in mucus all the time. Your food, when it goes in your stomach, it almost never touches the wall of your stomach because there's such a thick layer of mucus lining inside of your stomach. Why is that? Well, it's because your stomach has got a pH of about 2. It's really, really acid. And so you need to be protected from the acid by this thick layer of mucus. What are the functions of this simple columnar epithelium? Well, secretion. In the stomach, in addition to secreting mucus to protect the wall, it also secretes a bunch of digestive enzymes that start breaking down the food. Pepsin, pepsinogen, those break down proteins and so forth. That is caused by these enzymes that are secreted by this simple columnar epithelium. They do some absorption too, especially down in the colon. They absorb a lot of water. Basically, that's the only. My teacher used to always tell me that the colon only has 2 functions. One is to reabsorb water. And just as an example, we dump about 5 to 7 liters of fluid, of intestinal juice, out of our small intestine into our colon. 5 to 7 liters, quarts a day. But by the time it gets around our colon and is ready to go out our rectum, we might make 250, 300 cc's of stool a day. So we have reabsorbed all that water in the process of passing around through the colon. There's also some vitamins and stuff that get absorbed there. Okay, here's one of the 2 odd ones. Pseudostratified columnar epithelium. It looks like it's stratified. The reason it looks like it's stratified is because the nuclei are not all in one nice line. Okay? In simple columnar epithelium, the nuclei are always in one line. But in this pseudostratified ciliated columnar epithelium, which is what this really is, you notice that some of the nuclei are down at the bottom, some of the nuclei are up at the top. But the reason it's called pseudostratified is because every single one of these cells, even this cell here, actually extends to the surface. Okay? This cell goes all the way from the surface down to the bottom. Okay? So it really is simple epithelium, but it doesn't look like it. It's pseudostratified. Now, we call this pseudostratified ciliated columnar epithelium, PCCE. You can write that down because we're going to throw that word around, that set of initials around a lot. PCCE. Pseudostratified ciliated columnar epithelium. We call it ciliated. Why? Because it's got cilia up on top. Okay. Where do we find this kind of stuff? We find it basically in the respiratory system, in the trachea and in the bronchi. PCCE. Where do we find a non-ciliated pseudostratified? It says parts of the male urethra. It's a tiny little part. I would ignore that, sort of. I just think that's really splitting. So here's an example of PCCE. Here's the basement membrane down here. Here's the surface up here. These are cilia. You can't see it perfect on this slide. And notice that the nuclei are not all at the same level, even though it is a simple epithelium that's only one cell layer thick. Here's a better picture of that same thing. You can really see the cilia nicely up here on top. These are the little guys that are up there trying to push all that junk back up in your mouth from down in your trachea. Here's the basement membrane right here, and notice that the nuclei are at different levels. So what are the functions of PCCE? Well, it lines the upper respiratory tract, and the cilia sweep stuff back up towards the throat. So it's basically a protective type of epithelium. It does secrete stuff. It has a bunch of goblet cells in it that secrete mucus. Let's go back to that slide and see if we see any mucus cells. I guess I really don't. Okay, stratified. Let's talk stratified now. We talked simple, right? And there's basically four kinds of simple. There's squamous, there's cuboidal, there's columnar, and then there's pseudostratified. Okay, those are all simple epitheliums, because in all of those four, every cell goes from the basement membrane to the surface. Now let's talk about the stratified ones. Stratified ones, if it's stratified squamous, the top layer is the one we're going to name it by. The top layer is going to be flat. The bottom layers can be any shape. Just to kind of ignore the bottom layers in terms of their shape. What's their function? Well, if you think about it, to protect the underlying layers, you want to have a lot of layers of stuff up on top. So if you went like this to inside of your trachea, that thing would be bleeding and it'd be a mess. You'd scrape pieces of it off. But our skin, for example, is a bunch of layers, and they get more mature and they get drier as they come up to the surface. But we also have stratified squamous other places. But just think of this. Whenever you think about stratified squamous, think protect underlying areas that are subject to abrasion. Now, why do I mention that? Well, think about the inside of your mouth. Okay, it gets abraded. You're chewing up stuff. You're chewing up bones. You get rough bread or whatever in there. It's going to scrape the inside of your mouth. So you want to have something stratified in there to protect you. And indeed, that same thing holds true for every place where your body goes from the inside to the outside. In other words, the inside of your mouth, inside of your nose, inside of your vagina, inside of your rectum, anywhere where the inside comes in contact with the outside. There's a transition area that consists of stratified squamous epithelium to give it some protection, against abrasion. All right, here's an example of stratified squamous epithelium. Now this is what's called non-keratinized. Let me put that word keratinized out here to the side for a few minutes, okay? This is non-keratinized. So the inside of your esophagus, which is this tube that goes from your mouth down to your stomach, obviously, it's composed of a bunch of different layers. As you can see here, this is the basement membrane down here. It comes up in ridges like this. So these are all the epithelial cells from here, the basement membrane, to here, which is the surface. And you notice that the cells at the very top are very thin, they're flat, right? So that's why it's called squamous. If you had to name it from these guys down here, you sure wouldn't call it squamous, would you? Okay. You remember one of the characteristics of epithelium that I talked about early on, and I said there's not any blood vessels in it? Well, once again, if you look from down here to up there, no blood vessels. How does this area get blood? How does it get oxygen? How does it get glucose? How does it get all the stuff it needs? How does it get its wastes removed? Well, the blood vessels all live right in here. That's a blood vessel kind of caught lengthwise. Here's another one right here full of blood cells. The blood vessels always live in what's called the submucosa, right below the basement membrane. And so indeed, in a lot of these types of tissues, the submucosa will come and be heaped up like this, once again, to make more surface area so that more of these cells can get their nourishment from here. Okay? This is another example of the same thing. It's non-keratinized. Once again, notice how cuboidal the cells are down here, but by the time they work their way up to the surface, they are very thin. And when I say work their way up to the surface, that's exactly what I mean. The new cells are made down here. You can see some mitoses down here. See that metaphase right there? And this is where all the new cells are made, and then they gradually work their way up. And as they work their way up to the surface, then they just fall off. They fall off, even in the non-keratinized areas like this, which is like the esophagus. I just can't decide whether I want to throw this Apple Watch about 100 yards and never look at it again. It drives me crazy. But as you get older and you get deaf, it's nice to have something vibrate on your wrist to tell you your phone is ringing. Many times I've missed the phone call without this. Okay, now we talked about non-keratinized. And keratin is a protein that the cells accumulate as they mature, and it makes them, sort of turns them into leather. So the best example, and I guess maybe the only example I know of stratified squamous epithelium keratinized is the skin. Okay, skin is everywhere on the outside, and when you look at these under the microscope, you will always see this stuff up here on top. Think of it as the cornified layer, the corny layer, the flaky skin. That's what this is up here. As the cells are formed down here, and as they mature and come up through the layers, they gradually are processing keratin, becoming more and more keratinized, and that's why our skin on the top is dry. Okay? It's leathery, and it's stretchable. It does all those wonderful things for us. The only place you'll see this kind of appearance is in skin. Keratinized, stratified squamous epithelium. And I won't try to trick you, I promise. If it's keratinized, you'll see the cornified layer really well. And if it's not keratinized, it'll look like this. And it'll be nice and smooth on top. Okay, see the difference? Okay. So here's skin. We're going to do a whole chapter on skin here in a couple of weeks, maybe in a week, real quick. So what is skin's function? Our skin's function is mainly physical protection. It keeps bacteria out. It keeps heat in, keeps heat out. Protein, keratin is a protein that helps strengthen the cells against abrasion. So if you scrape your forearm, it's probably not going to cut it at all. But if you did that to your esophagus or any of the other areas that are non-keratinized, stratified squamous, you'd rip it and pull pieces off and it would bleed. Okay, next one is transitional epithelium. You're probably thinking, well, wait a minute, Sessions, why did you skip stratified cuboidal and stratified columnar? And you're going straight to transitional. Give you the answer in a minute. All right, here's that transitional one I told you is made to stretch and get smaller. It's always several layers thick. It's not single. It's not a simple. It's a stratified epithelium. The basal cells are usually cuboidal, and this is the key word. Remember this word when it comes to transitional epithelium. Remember the word dome-shaped. Okay? And what that means is that when the bladder is not stretched, okay, when it's not stretched, the upper layer may look like this. and every so often you might see a big dome-shaped cell. That's what that is. Now, when this baby stretches out, we have examples in the book and in the lab of stretched bladder. You won't see any of these because they will have stretched out. But that's the whole idea of them, is to allow them to stretch and make your bladder bigger so it can hold some urine. And the rats you saw, their bladders are really tiny. They don't ever get big because rats are just socially unacceptable. And that's really the only function our bladder does for us. It allows us to be socially acceptable and not have to pee every 5 minutes like rats do. So anyway, remember that word, dome shape. And let me just tell you that once again, this is not something that we're going to try to trick you on. Okay? If I'm showing you transitional epithelium, it's going to have dome-shaped cells. You're going to be able to see them. But you need to practice. You need to look at a bunch of bladder slides so you know what you're looking at. Okay? It's only found in the urinary system. These cells on top are flattened out when the bladder is all stretched out. What's its function? Simply to allow for expansion and recoil. Here's the picture. Here's the distended bladder. You notice all the cells are pretty flattened out. Here's the empty bladder with a couple of big dome cells up on top. I can't give you more hints than that. Okay, here's the two we skipped. Stratified cuboidal and stratified columnar. Now, why would we skip those? Well, first of all, they're very, very rare. They're only found in a couple of places in the body, and I doubt I'm ever going to ask you. I might. But they're very rare, but they're really obvious when you look at them. Because although they're stratified, and those other examples of stratified tissues, you know, I showed you they had like 20 or 30 layers. Well, the most these ever have is 2, okay? So whenever you see stratified cuboidal, it'll be 2 layers, 2 cells thick. Whenever you see stratified columnar, it'll be 2 cell layers thick. So that makes it pretty easy. What are the functions? Mostly it's just these are cells that secrete, found in some sweat glands and mammary glands. Example, here's an example. This is mammary tissue. These are mammary ducts. These are the ducts that run all through the breast. And notice that each duct, here's a duct right here, and you notice it has 1, 2. 1, 2. 1, 2. Here's the lumen in the middle. 1, 2. I need to do that a couple more times. 1, 2. Those are stratified cuboidal. Stratified cuboidal again. There's another sweat gland. Stratified columnar also is just found in a couple of places. Well, I should back up a little bit, but the thing you just need to remember, the one that this one gets tricky with is the difference between stratified columnar and between pseudostratified columnar. Remember, pseudostratified, the nuclei are kind of scattered around, but every cell goes from bottom to top. The difference in this one is that it's 2. 1, 2. So there's 1 line of nuclei, 2 lines of nuclei. 1 line of nuclei, 2 lines of nuclei. Get it? Okay, so stratified cuboidal and stratified columnar are only found 2 cell layers thick. Another one here, stratified columnar. I think you could confuse this one here with stratified cuboidal, right? Because these cells, and they're taller, but not significantly. Okay. Hey, that's it. That's epithelial tissues. So we're going to start today in lab, and we're going to start looking at a lot of these. And the things that you need to be looking at is trying to remember the characteristics that I told you here. That's going to be, these are the hints that are going to allow you to tell the difference between these tissues. Call me over. I'll be happy to look at them with you. All right, let's talk about connective tissues. That's the second of the big four types of tissues. Uh-oh, somebody will be mad at me. Wrong kind of pen. I should just use magic marker and use that white wall over there. They really do this. Oh my God. All right, connective tissue. So what does that mean, connective tissue? Well, it means simply it connects things together, okay? And so it connects the epithelium. The epithelium, remember, is going to be the stuff on the outside, on the top, and on the inside, and all the stuff in between is going to be connective tissues. So some examples here are bone. Bone is a connective tissue. It supports you, it holds you together. Store energy, fat. Okay, fat, it fits in between everything. We all have fat. We have it all over the place. Some of us have more than others, and we're quite proud of it. Another connective tissue is blood. Blood may not seem like a connective tissue, but if you figure it fills in the spaces in between, and it also carries a lot of stuff around for us. It carries sugar to the cells. It carries oxygen to the cells. It takes wastes away. Remember that connective tissues never, ever have contact with the environment. The only time you'll ever see connective tissue is when you dissect something or when you're in an operating room and you see the inside of folks. That's the only time you'll ever see connective tissue. All right. This is the most common kind of connective tissue. It's called connective tissue proper. I'm going to give you several names for this. And just kind of remember that they all mean the same thing. This is connective tissue proper. This is what it looks like under the microscope. And what are the simple things about it? Well, first of all, notice that most of it is just clear. That's called the ground substance, or just the fluid that's in there, interstitial fluid. There's always some collagen fibers. These pink wide fibers here are called collagen fibers. Collagen fibers is what makes you tough. That's what collagen makes up tendons. Collagen makes up surroundings of muscles and so forth. There's also another kind of fiber in here called elastic fibers. Elastic fibers are the little thin black ones. Okay? What's the good thing about the elastic fibers? Well, they stretch back. So that's why when you pinch your skin, it doesn't just stay there when you pinch it. It snaps back to normal shape. The most common kind of cell found in connective tissue is the fibroblast. Fibroblast. Well, duh. What do fibroblasts do? They make fibers. Okay? So, let me just tell you right now that when you see fibro, think fiber, when you see blast, any kind of blast, that means an immature cell. In other words, or it's the cell that makes stuff. Okay? So fibroblasts make fibers. Fibrocytes are fibrous cells that take care of the fibers. Osteoblasts, osteobone blasts make bone tissue. Osteocytes are mature bone cells that just take care of the bone around them. Chondroblasts make cartilage. Chondrocytes are mature cartilage cells that take care of the cartilage that's around them. Okay? So all of these words get a lot easier once you kind of know what the syllables mean. Another kind of cell that we see a lot of in connective tissue proper are called free macrophages. What do macrophages do? What? They eat up bad stuff. Yeah, they're the garbage collectors. They come around and eat it up if you don't want it. So if a cell dies, macrophages come in and eat it up. Now, this is another picture of the same thing, only this is just a schematic drawing. And this has got a few more kinds of cells in it. And when you look at it under the lab, you'll see most of these. Most of them you'll be able to identify. Here's the ones I think are important. First of all, the collagen fibers are the long, stringy ones. Collagen is kind of cool. It's very strong, but it's only strong in the one direction. Isn't that a boy band? That was bad. It just hit me suddenly. So collagen fibers pull in the direction in which you're pulling. And so when you look at things like a tendon under the microscope, you're going to see that all of the collagen fibers are organized in the same direction. So they're really strong when they're all lined up just right like that. In this example here, they're not. So these are some fat cells. We call fat cells adipocytes. Adipose tissue is fat tissue. Adipocytes are fat cells. We also call them signet ring cells. This is sort of a signet ring here, and if you look at it from the side, you see that it's got a great big hole in the middle. That's like the fat here, and then on one side over here, it's got the nucleus and the organelles and stuff. So basically, a fat cell is nothing but a big bubble of triglycerides. Fat. All right. Notice there's some blood vessels with blood cells in it. There's plasmacytes. There's melanocytes. What do you think melanocytes do? They make melanin. They do not make melatonin. I just heard that. Melatonin is a hormone secreted by the brain to help you sleep. Anybody use melatonin to sleep at night? I do. It works really good. It's a natural hormone that makes you sleep. But that's not melanin. Melanin is the dark stuff. Okay. So here's fibroblasts. Here's a free macrophage. Occasionally you'll see a mast cell. Mast cells have a whole bunch of dark-colored inclusions in them, little vesicles. They contain a bunch of heparin and histamine and stuff like that play a big role in allergic responses. And you notice that most of it is this ground substance or just a clear fluid background. Now, have you ever seen connective tissue like this? Well, you don't know you have, but you have. You know, the other day, some of you, when you would pick up the skin, you lift it up, there's that kind of crinkly stuff between the skin and the muscle underneath it. Well, that's all connective tissue. That's what you're looking at right there. And I know when I was learning how to do surgery, I had an attending surgeon who would say, "Come on, Sessions, cut the crinkly stuff." And we would just always dissect in the lines where there was crinkly stuff, because that way you didn't get into that organ, you didn't get into that organ, you just divided it through the connective tissue to keep separating things away the way you wanted to. Anyway, there's 9 different cells that occur in the connective tissue. I think I've already mentioned them. I'm just going to run through them again. Fibroblasts are the most common. They make the fibers. They secrete these proteins and hyaluronin, which is kind of the cement that sticks it all together. Fibrocytes are also important. They maintain the fibers. Macrophages are the large amoeba-like cells. Adipocytes are the fat cells, and mesenchymal cells. These are kind of stem cells. When I say the word stem cells, what does that mean to you? Growth. What? They don't know what they are yet. That's a very good way to describe them. They don't know what they are. And the nice thing about having some stem cells around is if you need more fibroblasts, they can turn into fibroblasts. If you need more macrophages, they can turn into macrophages, and so forth. So stem cells are important. That's basically the mesenchymal cells. That's another word for them. Melanocytes make the brown pigment melanin. Mast cells stimulate inflammation, so they release histamine and heparin. I think I'll just... Lymphocytes and basophils, which were on the last slide, those are just blood cells. I think it's important that you realize that some of the white cells, some of the blood cells don't stay in the blood. They don't stay in your blood vessels all the time. They actually go right out through the walls of the blood vessels and get out into other things. Anybody ever had a little abscess or growth, pus pocket somewhere? And that's a pus pocket is nothing but a bunch of bacteria, dead bacteria, and white cells. Those white cells were in your blood, but they went out to go and try to attack and fight the infection. If you ever got a little splinter under your skin, there's just tons of white cells that work their way in there trying to digest and make that splinter go away. Collagen fibers, you see tons of them in tendons and in ligaments. So when these football players tear their ligaments, they're basically tearing a whole bunch of collagen fibers that were running in the same direction. And the last one, that is the third kind of fibers that I didn't mention, but you need to understand, are what are called reticular fibers. Reticular fibers, what does that mean? Well, reticular fibers are like a network of fibers that are attached to each other that kind of do like this. And they tend to create organs and sheaths around organs, and they create the organs themselves like the liver. You guys saw the liver the other day. You notice how the liver kind of has a shape to it, but yet if you take your fingers and pinch it, it just like all falls apart. The thing that's holding the liver into the shape that it is are reticular fibers. Now the tricky thing about reticular fibers, pay attention to this, is you will never see reticular fibers on a regular slide. You will never see reticular fibers on a regular stained slide. Okay? So we have a bunch of slides over there that say reticular fibers, and they're stained specially for the reticular fibers. And usually the reticular fibers appear really dark, and they're all branched, and they're just designed to hold things together. They resist force in many directions. They stabilize a lot of these cells, and so forth. Elastic fibers already mentioned. Ground substances, clear fluid. Okay, I'm going to stop right there. Okay, so we'll meet in lab in a little bit, and we'll start looking at some things under the microscope. I'll take a minute to jump online and see if I can make these videos play.
I appreciate that.
Sometimes the computers here at PCC do not have things on them like, Silverlight and like some of those plugins. Melissa knows how to make it work, though. If she's here today, I'll call her to.
I really don't think it's my internet connection or my computer, because I just bought a brand spanking new Dell i3 Intel processor, and I have.
It also means you don't have all of the